Hi friends, in this video we will discuss about the recombinant DNA technology. So what is this recombinant DNA technology? It is often known as rDT. So it is a new field in science. So it does the alteration of the genetic material. So what is the genetic material? It is the DNA. So it alters the DNA. It alters the DNA so that a better DNA is being generated. So this technology is often referred as genetic engineering, as we engineer the DNA. So what does it actually do is that it brings together many genetic materials. It can be two, it can be even more. It brings together these DNAs and joins them. It joins each DNA so that a new DNA is being formed. So this technology is called recombinant DNA technology. So now let us discuss a simple definition to the recombinant DNA molecule. So a recombinant DNA molecule is a molecule that is formed by joining two or more DNA segments. So two or more DNA segments are joined together, and these DNAs are from different organisms. So two or more DNAs from different organisms are joined together to produce a molecule called recombinant DNA molecule. So now, to explain the recombinant DNA technology, let us take an example. First, we take a DNA, and this DNA is from human, and we select a human insulin gene from the DNA. So we have a small segment of DNA in our hand, and now we take a plasmid, and a plasmid is an extracellular DNA that is present in a bacterium. So we have two DNAs in our hand. One is human insulin gene, and the second is the plasmid from a bacterium. Now, using the tools in recombinant DNA technology, we join these two DNAs together. The two DNAs join together to form a recombinant DNA molecule. This recombinant DNA molecule, which has a human insulin gene in them, is inserted back to a bacterium. So now the bacterium is having recombinant DNA. So we call the bacterium a recombinant bacterium. A recombinant bacterium is added to a fermentation tank, and in the fermentation tank, this bacterium produces insulin. This insulin is being harvested from the fermented tank. The insulin is being purified and later is commercially distributed as a medicine. So this is how a recombinant DNA technology works. The same steps are followed by all the products that are produced by recombinant DNA technology. In recombinant DNA technology, there are different tools used. One of them is restriction enzymes. So these restriction enzymes cuts the DNA. So these are the enzymes that are used for the cutting of the DNA. So a second tool that is used in recombinant DNA technology is ligase enzymes. So these ligase enzymes are used in joining together the appropriate fragments. So for cutting of the DNA, we use the restriction enzymes, and for joining the DNAs, we use a ligase enzyme. So first what we do is that we cut a DNA. We cut a DNA as well as we cut a plasmid. So what is a plasmid? A plasmid is extracellular material that is present in bacteria. So we cut these both DNAs and we join them. We join them by a ligase enzyme. So after which they are cut, they are joined. So this joined molecule is called the recombinant DNA molecule. After which we have got a recombinant DNA molecule. This molecule is then introduced back to a living organism. This living organism can be bacteria, for example, and in this bacteria, this continues to multiply. So, this multiplies and makes copies. So, this is called a technique, gene cloning. So, this technology is called recombinant DNA technology or gene splicing. So, first what we do is that we take a human cell, and from a human cell, using restriction enzymes, we cut and take a gene. So, for example, we took an insulin gene. From a bacteria, we take a plasmid. So, this plasmid is also cut using a restriction enzyme. And this insulin and the plasmid is joined together, and they are joined together by DNA ligase. And the structure which is formed is called recombinant DNA molecule. These recombinant DNA are later taken up by a host. So in this host, they start to clone, that is, multiple copies are produced, and they start to produce the insulin, and hence we get insulin. This is how the insulin is being produced. So now we will discuss the important steps in recombinant DNA technology. First is the isolation of the genetic material. Second, it is the cutting of DNA at specific location. Third is the recombinant DNA formation. Fourth is the cloning of the DNA. So now let us discuss the first topic, which is isolation of the genetic material. So for a majority of organisms, DNA is the genetic material. So these DNAs are present inside the nucleus. That is, inside a nucleus, there are chromosomes, and inside the chromosome, it is DNA. So to isolate the DNA, we have to first break the cells. So we use many enzymes. For a bacteria, we use lysozyme. For plant, we use cellulose. And for fungus, we use chitinase. So in order to get a pure DNA, we also need some macromolecules like RNA, proteins, and polysaccharides. So these are isolated by treating with appropriate enzymes. So the isolated genetic material is called the foreign DNA. So this foreign DNA is incorporated with the bacterial plasmid. So first step in recombinant DNA technology is the isolation of DNA. DNA has to be isolated first. So DNA is present inside the nucleus. So inside the nucleus there are many chromosomes, and inside the chromosome it is DNA. So in order to get the DNA, we first have to treat the cell. That is, the cell has to first break in order to get the DNA. So in order to this, we use lysozyme. This lysozyme breaks the membrane of a bacterial cell. And for a fungal cell wall, we use chitinase. Chitinase is being used for losing the cell wall of a fungi. Next step is the cutting of the DNA. So DNA has to be cut at a specific site. So for this function, we need a restriction enzyme. So a restriction enzyme is often known as molecular scissors. It is thus known because its function is to cut the DNA. So it cuts both the foreign DNA as well as the plasmid DNA at a specific site. So these sites are palindromic in sequence. So what does this palindromic mean? It means that a sequence is the same read when it is forward and it is backward. That is, it can be read the same when it is read from forward direction and when it is read from the backward direction. For example, if we take the English word madam, it is read the same from right to left and from left to right. So such sequences is called palindromic sequences. So the step 2 in recombinant DNA technology is cutting of the DNA. It is cut using a molecular scissors. So a restriction enzyme comes to a DNA and it cuts the DNA at a specific site. This diagram shows a palindromic sequence. A palindromic sequence is a sequence of DNA in which one strand, for example, if we take 5' to 3', the sequence is same in the other strand which is running in the same direction, that is from 5' to 3' of the other strand is the same of the previous strand. So such strands are called palindromic sequences. That is, when the reading orientation is kept the same, they have the same sequences. So such sequence is palindromic sequence. So now we will discuss the third step, which is the formation of recombinant DNA. So as we discussed earlier, we know that a piece of gene or DNA is called a foreign DNA. So a foreign DNA is combined with the plasmid DNA. So both of them, the foreign as well as the plasmid, is cut at a specific end, leaving a sticky end or a blunt end. So they are joined together by ligase enzyme, and hence the formed structure is called recombinant DNA. So as we discussed earlier, a restriction enzyme can cut a DNA in two patterns, either in a sticky end pattern or in a blunt end pattern. So sticky ends are the cutting that leaves a overhanging, as you can see in the diagram, while the blunt ends do not leave any overhangings. So hence, sticky ends are also known as overhangings, whereas blunt ends is known as non-cohesive cutting. So example of sticky end is EcoR1, whereas example of blunt end is EcoR5. So what is a recombinant DNA? On joining the DNA fragment with a vector, for example a plasmid, we get a recombinant DNA. These two pieces of DNA are joined together by DNA ligases. The two DNAs having each a sticky end, they both are complementary to each other. They are ligated using a DNA ligase, and the structure thus formed is a recombinant DNA. That is, it has two DNAs joined together. The next step and the last step in recombinant DNA technology is cloning of the DNA. The recombinant DNA has to be cloned. So, we introduce it into a rapidly dividing bacterium. So, for example, we take a E. coli. So, the process of introducing this recombinant DNA into bacterium is called the transformation. So, these bacteria, for example, if we are taking a E. coli, these takes the DNA from the surrounding, and this is through a process called transformation. So the bacterium which has the recombinant DNA now is brought to a nutrient medium, and in this nutrient medium, these doubles. In every 20 to 30 minutes, it doubles and produces millions. So, hence, millions of copies of recombinant DNA is formed. So, a recombinant DNA is taken up by a bacteria through a process of transformation. And after this process, the bacterium reproduces It reproduces and produces millions of copies of recombinant DNAs. So now let us revise the important steps of recombinant DNA technology. First is isolation of DNA and so that we get a foreign DNA. After we get a foreign DNA, the insertion of DNA fragments into a suitable vector. That is, a vector used here is usually a plasmid DNA. So a foreign DNA is inserted to a plasmid DNA. The next step is introduction of the recombinant DNA into a bacterium. And this is done by a process called transformation. And usually we introduce it to E. coli. After which it is introduced, it is multiplied. It is multiplied so that multiple copies of recombinant DNA are obtained. So this is about the recombinant DNA technology. Hope you understood the concept. For more such videos, do subscribe to our channel. If you like the video, please like and share with your friends.